a Chinese company that took advantage of a virus that is circulating, right? The SARS-CoV-2, inactivated the virus. This is very simple concept. And you give to animals or give to people and see if it's going to be protected. Basically, here you see the empty shell of this of the vaccine. So, but it's not only that, right? So we have two things here to discuss. One is the immunology inside of this empty little bottle. And the second is, why am I holding a vaccine, very simple, made by a different place to be injected in our people? I want you guys to think about that, right? Because actually, this is for a young generation as well as to the old generation. So the old folks, us, we have to think about that as well and stimulate young generation to be aware of what's going on in terms of science and science-based problem resolution. All right. So I'm gonna turn on now and share a few slides with you. That's gonna be what I'm gonna be speaking about today. So, Hai has my WhatsApp if you guys have any problem with the. All right, so you can call me or anything like here because the internet can be kind of weird these days.
All right.
But so that's gonna be my title. I'd like to show you a little bit of what is like to ride the immune system with the whole deal of vaccines, right? So we're talking with Hai, Bonnie, and Nadia before. So I wanted to put a little map for you guys here. So we are located here in the state of Santa Catarina. Here's Colombia. I hope I can visit you later. So I wanna be surfing right here. Oh, right here. Hai can take me to the Caribbean Sea. Hai and Beth. So we're gonna go here. And the second talk that I'm gonna give to you after the pandemic. So, but we're located here in the Santa Catarina state and in a little island about 100 kilometers wide length. So that's the island called Florianopolis. We call the magical island or Isla de Magia. Basically, we're here at the university where the heart is. So I'd like first to give many thanks to our team here. We have many collaborators, many people working in these projects that I'm going to be talking to you about, and institutions of Federal University of Santa Catarina, where I am now, Universidade Federal de Minas Gerais, Universidade Federal do Rio de Janeiro, no Hospital Cross the Street, Instituto Butantan, that I'm sure that you guys are aware of, one of the big vaccine producers here in Brazil, University of Cambridge in England, in the UK, and the Karolinska Institute in Sweden. So basically, I want to say thank you to these guys. So what is immunity, basically? So here you can see the experiments, very early on experiments that the paper was published in 1890 by Baron in Kitasato, where they took guinea pigs. Let me try to do this here. Where they took guinea pigs and inject them with a toxin from bacteria. And they took the serum from this guinea pig exposed to this toxin, and they inject in different guinea pigs. And later, they challenged these animals with the bacteria that causes disease, in that case, diphtheria. And you can see when they did that, some of the guinea pigs were protected. They didn't have disease. And they claim because they gave them the serum that's been from animals pre-exposed to the toxins. And also what they also did was that they gave a different toxin and challenge with the same bacteria as the other group, but they still were got disease. So that's the first one of the first concept of serum specificity, and they called immunity. Though that's development of immunity. They won a Nobel Prize, but I want to just to on I think was the first Nobel Prize on medicine and physiology by the time. But here you can see very simple concepts, and people don't know anything about antibodies, about what was going on in the serum, and how they could develop this so-called immunity. But I want to just to introduce you to so we can talk about um, more. We can talk more about immunology using this concept. So here, how it's gonna go. So I'm gonna give you a brief review of the immune system, so we can try to speak the same language. I'm also gonna put a slide with the vaccine strategies from an immunologist perspective. I'll give you a little bit of challenges that were exposed here in Brazil with the pandemic. I'm pretty sure that in Colombia it was not different. And opportunities that we can think as a community, how we can tackle problems like pandemic, or even day by day problems that we can solve using immunology or science for that matter. Um, so I'm gonna focus on human immune system, what is known about it. Basically, don't know that much. All immunology foundations for 100 years or more were basically performed in experiments using animals, experimental animals, no human animals. For example, mice, guinea pigs, monkeys sometimes. Um, so all the organisms pursue some sort of alive organisms pursue some sort of immune system. For example, plants, they have immune system. Flies, even bacteria also have adaptive immune system called CRISPR, for example. But I'm gonna concentrate it on human immune system. I hope you guys are listening well. There, I have. Um, so what we so so we can divide the immune system in first part would be recognition. Let's say innate immunity would be a very conserved manner that the immune system recognize molecules or components present, for example, in infectious uh, pathogens. So recognition is a key is important for the immune system. So you have to be able to recognize and to discriminate what is self to the non-self. That's what the immunology dogma presents to us. So in the recognition, um, we know that many receptors present in the cell surface or intracellularly, they recognize different components from of or produced by pathogens, or even from your own body. Right? And so these would be what we call recognition. The other thing that's very important after recognizes that this immune system does is the migration. So cells, they have to be able to recognize, transform the information that something is wrong with your body and migrates to different tissues. You can see a lymph here in this person, a lymphatic vessels that is draining virtually all kinds of tissues. So the cells or the immune system cells, they have to have the right fit of receptors. They can go up or down, left, right, and they know what the danger is. So basically, this is a very important process. And the language that immune cells does for that, is that receptors that they express. So you have, for example, T cells that express receptors that will enable them to migrate to the skin or to the intestine or whatever tissue you may think of. So migration and information is very important. The other aspect of the immune system, very important, is the selection process. So one might say that you have a Darwinian selection process when cells they interact with the antigens, they are picked, and in doing so they activate and after activation they expand. And here, for example, you can see a dendritic cell, which is one of the antigen presenting cells. So they take up antigens, and you can see more closely here. They present antigens through certain receptors, for example, MHC, and they are recognized by T cells. Here you can see the TCR receptor, which is specific for the MHC plus the peptide. So this little thing here, this little dot you see here inside the MHC class 2 is an epitope or peptide. It's very small. They can be 7 to 20 amino acids. And you have other receptors which are molecules that they are called stimulators. So after the dendritic cell presenting antigen, one of the cells would, or a few cells would recognize uh, this antigen and will divide. So we'll call expand. And then we have something called adaptive immunity. And with adaptive immunity, after a while, the cells contract, which means many of the cells die out because they do not encounter antigen in the periphery sites. So they simply go into apoptosis. So that's why you have a contraction. And you can see here in this figure, we have in the y-axis a number of cells. In the x-axis, we have a kinetics, a time. So cells proliferate, they reach a peak, and then they contract. So you have decrease of these cells. So you also can see here that at later time points, we have a few cells circulating. So these green cells means that they have the same receptors, that were selected by the antigen that one would be exposed to. And then you have the thing that we call clonal selection. And these cells that remain and they're circulating, we sometimes call them memory cells. So memory cells would be an important aspect of adaptive immunity in humans that would allow us to explore and take advantage in the building, in the generation of vaccines. So I took this from translated versions in Portuguese from Abbas. I wish I could find the Spanish one. But that's the concept that in the book, probably many of you already read about it. You have innate immunity, la inmunidad innata, acá, and the microorganismos, when they encounter the epithelial barriers such as skin, some of these organisms pass can invade these barriers, and they're gonna get picked up by phagocytes like phagocytos and células dendríticas. Okay, and then you have all the receptors in the cells trying to fight the invader microorganisms here. And this happens all the time, many times a day. All the time we're exposed to these little microorganisms that are out there, and our innate immunity, it's engaged and activated. uh, in parallel, I might say, what's going on, as I told you before, is that the dendritic cells migrate, or antigen migrate to the lymph nodes. In the lymph nodes, you have antigen presentation. For example, the antigens can be picked up by lymphocytes, by B lymphocytes. B lymphocytes are wonderful cells. They're amazing. They make antibodies, and you can see the little Y-shaped molecule here. I'm gonna talk you in a moment. And these antibodies recognize pathogens. Um, Also, dendritic cells or even B cells, they present antigen to T cells, to lymphocytes. Los linfócitos T, que también tenés receptores acá, the TCR, they will divide it up, and you have the effector T cells. And they recognize cells that had phagocytosis pathogens, and they, for example, they can stimulate the killing. So this part here is also called adaptive immunity, and that can happen in after days after infection. So once you're exposed to the microorganism, let's say an inactivated microorganism contained by a vaccine, days later, you'd see adaptive immunity being triggered. The thing is, Abu Abbas draw this cartoon here, which is a scheme, right? Draw, and so many antigens we already know about it, but the thing is, we didn't know what's going on with the SARS-CoV-2 in terms of this cartoon. So if we were to generate a scheme with this kind of information for SARS-CoV-2, how would that be? Think about that. If you want to, if this microorganism is a coronavirus, a SARS-CoV-2 that is infecting people in Colombia, for example, and you study all these cells, all this process coming together, and you actually can describe day by day, following weeks, how would that be? What kind of scheme would you make, right? Do you have data on this? Do we in Brazil have data for the Brazilian court on exactly these kind of things? The answer you already know. We don't know. So classical aspects of adaptive immunity. So in adaptive immunity, we have DNA rearranged receptors. So that's why in mammals or in many vertebrates, you can select different receptors. And these can be 10 raised to 15 different possibilities in the DNA. So we also have specificity. We also have the discrimination between self and non-self, diversity, and memory. Those are key concepts for the adaptive immunity. For the innate immunity, which here in the lab we have been studying quite a while, We have all this concept. We have specificity, we can discriminate self and non-self, we have diversity, and we also have memory, but the receptors are not rearranged. Um, and I wanted to also show you a picture of, let's say, a molecule that many of you already know. It's called immunoglobulin, or the antibody. There's a Y shape so the structure has been solved by Eldman in 72, so many years ago. And it's shaped in a way that it can bind the pathogens. Here you can see this region is the region that binds the pathogen, this and that region. We also call this region FAB. And also there's this region called FC, which interacts with many enzymes or complement or cell receptors. So this molecule is very flexible. It's very important in the immune systems. That's highlighted, for example, for when you have examples of people, now human beings that are genetically defective in making certain antibodies, and they have uh, more risk in getting infectious disease, certain infectious disease. And I'd like to call your attention for this guy over here. So this is a SARS-CoV-2, the example, the structure, just a cartoon, but it's this. Let me open. I wanna go here again. So sorry. So here you see uh, uh, an area of the SARS-CoV-2, the coronavirus. In green, you see a model of the spike protein, which is apparently is a very important protein for the infection cycle of the coronavirus. And in those all these colors, pink, red, over here, you see antibodies. So, and though the scale here is the scale supposed to be what's going on in real life, what we call real life, right? So, and then you see the coronavirus and the molecules going all crazy, and some of them are very good to recognize spike, but some of them do not recognize spike. So, and that's a molecule called the antibody that we discussed before. So how can you make vaccines? You know, what's going on? How can you actually think on a vaccine, right? So I would like to advise everyone. First, we have to know our environment. What's going on in the environment in terms of our what's going on in our place, our community, our city, our country. Disease mechanisms. If you understand disease, the mechanism by which disease is produced, it helps you when you're generating vaccine. Routes of infection, how a pathogen infects your body, that's also helps when you're thinking on vaccine. The pathogen life cycle is important. Protective immunity, how the immune system protects you against that pathogen. This is very important when you're thinking on a vaccine. Epidemiology and vigilance. Where is the pathogen being most spread? How does it spread? Who spreads it? For example, super spreaders are the people that spread the maximum of pathogens. Technology, what kind of technology do we have to make vaccines? Production and distribution, this is very important. you have to be able to produce the vaccine that you're proposing, and you also have to be able to to rapidly distribute this vaccine. So, and of course, there are many other aspects that you can take into consideration over here, but I'd like to highlight this one here so we can talk about later on these. And the question that people always ask me is, how can immunologists contribute on these on the pandemic or the vaccines or whatever? So for vaccines, for example, immunologists can contribute to characterize engines. So because, you know, if you think about the receptors that are being engaged during engine recognition, you can know more about the engines. Adjuvants, all the vaccines, they only work when you have adjuvants. So immunologists are very good on adjuvants research. Study immune responses. So take patients and analyze what they're going through in terms of the cytokines, the receptors, and the mind as well. Um, Screening and assays. So immunologists can also build up assays. For example, ELISA kits. So you can study immune response, you can study vaccination. Developing animal models. So how can you translate what's going on in humans to animal models? Analysis of the data. Vaccination strategies. What kind of population you can give the vaccines first, and much more that we can think about. We can think of type of vaccines. So we have all this type of vaccines, but we can create even more. I'm gonna tell you the classical. I just put it a list of the classical. So we have live attenuated, we have inactivated, replication defective pathogens, subunits, conjugated, viral vectors, and nucleic acids. What's going on more recently here. So I'm not gonna talk to you about all the strategies. I picked two. More recently, you guys have been listening about the mRNA vaccines. So the way they do it is that you pick an an antigen that you think, or it's an antigen that's involved in some sort of the pathogen life cycles. Let's say here you can pick a antigen from the virus, from the SARS-CoV-2 virus. And after that, you can bioengineer your RNA sequence. And then you have your RNA sequence. And you can synthesize that. And then you have your RNA sequence, and you make many, many copies of this RNA. And you do a linear DNA. And after that, you have many, many copies of RNA, purified RNA, and you formulate. In this case here, you're seeing lipid formulation, which are being used by Pfizer and Moderna vaccines. They call LNPs. So you try to protect the RNA and also make the RNA deliverable to the cells. You have after that to produce, scale up, put in the little bottles, and intramuscularly inject into people. So a lot of molecular biology is being done here, right? That's great. But before you do this molecular biology, you have to pick the engine. Which engine would you pick? And that's what immunologists can come in. And microbiology and biochemistry, and of course, I'm not saying that immunologists does at all, but I'm saying that this part is very important. And I like also to call your attention that if that mRNA vaccines really work, that we think that's working, it means that it's easy to make new vaccines based on the molecular biology steps. So if that's true, we have a very nice tool in hands to make uh, vaccines in a way so rapidly that we never done before. Okay. Uh, so you guys can look at this paper as it was a review published in Cell, just published in Cell. You also can look how they are making these vaccines and so on. We have experience here in the lab that I'm gonna discuss with you later. Just to remind you, here's the life cycle of coronavirus based on many decades of research. So coronavirus didn't show up out of nowhere, right? It was not like, we don't know, but it was not like, apparently it was not like some extraterrestrial being came into a little ship and put the coronavirus here. And we didn't know anything about it. No, we knew a lot of stuff about coronavirus based on research from the starting in the 60s. So the coronavirus needs a spike. There's a protein in the surface of coronavirus that makes a trimer. So they have to stabilize the trimer, and it goes inside the cell. These another receptors are being evaluated for the SARS-CoV-2, and in the cells there's its RNA genome positive sense that it's important to make all the virus protease and all the proteins from the virus, which then produces many of the capsids and so on. So basically, you have this virus being infecting through the apparently that's what we know so far, infecting through the aerosol routes, replicates inside the bodies. Many people will still asymptomatic and others have symptoms. So that's what we know about the virus. And what immunologists do, they look at the antigens that are important for entry, for example. So for 1 year, people have been looking at the spike because the spike was first demonstrated to be important for the viral entry. But after 1 year of the pandemic, we are and others are looking for different proteins, such as this protein called N protein from the nucleocapsid is important to make to protect the genome. Here you can see the N protein, the nucleocapsid on your right. We don't know what we don't know is if you have immunity, or if immunity is important, or immunity against the N protein is important to protection in terms of the virus infection, right? But what we know here, you can see the beautiful viral particle, and many proteins. You can see better the spike proteins and also the nucleocapsid, these proteins that inside the particle. And I draw you, not I didn't draw. I pick it up from some internet, from the Google. So you have a spike and you have the receptor. The ACE receptor is expressed in the cells that it's used by the spike or by the virus to get access into the cellular cytoplasm, right? So the spike is important here. So what we know is that if you have antibodies that block this interaction, you block virus entry and so on, and all the consequence of the virus entry and replicating the cells. So in the beginning, people looked at spike as as an important target, and all the vaccines are built up under this idea, except the vaccines with looking at the inactivated SARS-CoV-2, which will have the CoronaVac vaccine and one of our Indian vaccines. So, and then we decided to approach the one kind of vaccine, because we knew that all companies and governments around the world would be putting a lot of money to develop the vaccine that would be easier. For example, the inactivated vaccines or expressed spike proteins. And also the Pfizer and Moderna, they threw a lot of money on the mRNA vaccines. So since we're working here in the lab with the microbacteria and the BCG, which is the Bacillus calmette guerin, and our group had a lot of experience with the vaccination using BCG, we decided to use BCG as a vector of the coronavirus engines. So, and we, together with a very amazing team of collaborators here in Brazil and overseas in Europe, we decided to pick a few engines and construct and make constructs, uh, in that case, a chimeric between RBD and a portion of the N protein, and make a recombinant BCG that could stimulate both T lymphocytes and B lymphocytes to make antibodies and to make T cells against the SARS-CoV-2. So if you guys are curious, you can look at the publication from T Immunology by our colleague Sergio Oliveira at the Federal University of Minas Gerais, and we found a peptide in the end, which apparently through bioinformatics, it's a good stimulator of T cells and B cell response. And so what we are doing right now in this project is we're making constructs. We've been with the Butantan Institute, Luciana Leite and her group. We've made about 30 constructs of BCG, and they're plating out all the BCG to get the best BCG that expressing high level of these targets. The thing about that, when we knew already, is that BCG, it's kind of weird in terms of the antigen acceptance. Sometimes it goes really fast, but sometimes it gets really hard. And for the coronavirus antigens, it's getting really hard. So while we are waiting for these constructs to be ready to go, which are ongoing experiments right now, we're working on a platform here in the lab to overcome certain problems. First, we're producing different SARS-CoV-2 proteins, expressing these proteins. Second, we are also building up a kit, an ELISA kit ELISA assay to be able to evaluate immune responses from the candidate after candidates and injection. We are also setting up a neutralization assay, which takes into account a pseudotyped virus, which has the spike protein on it, and we can mix these guys, these pseudotyped virus with serum from people that have been vaccinated or sick, and we can measure the neutralizing antibodies against SARS-CoV-2 in these assays. And third, we decided also to begin with a platform, a strategy, which is based on RNA, on mRNA, aiming a best or optimal immune response. So, but the concept here, I want you guys to look at. The concept here is that if you want to stimulate T cells and B cells raised against the same epitopes, You have to give the right engine. Basically, we wanted to take advantage of people here in Brazil that have been vaccinated with BCG when we were newborns. And these people, they have the memory cells against major BCG epitopes. One of them is antigen 85. So basically, if you've been vaccinated with BCG, you have circulating on your body T cells against uh, the BCG, right, against the epitope. So what we're doing is we're making an mRNA, a hybrid mRNA, from using the spike of a variant of concern. In our case, we took the P1, which is circulating Manaus. Using this spike coupled with a peptide from the antigen 85B. So basically, when we're injecting this mRNA, we're telling the cell to make a chimeric protein. And we also put some of the things in the recipe as well that can cleave these proteins once they're inside the cells. So this chimeric protein would be presented to T cells and B cells. And then we're hoping to have now to boost the response, since you already have T cells which are specific to the antigen 85B. So basically, you're taking a ride here. Take advantage of the T cells that have been circulated that are already activated, and we're telling them to become also activated against spike. So basically, that's the whole idea here for this cognate strategy that we're implementing. That's the first, and we wanted to make sure that we're giving these target to the right cells, which we think that the nasal structure, since the coronavirus is spread in the aerosol, so we want to be able to localize the immune response in the mucosal respiratory system. We hook up with a group of engineers, chemistry engineers here at UFSC recently, and we want to make a powder to deliver our RNA if that's possible, right? But these are also ongoing experiments. I hope I can travel to Colombia and we can talk about more when we have these results after the pandemic. And I wanted to end with the two slides. Basically, so I wanted to, I threw something here in these slides which concerns all of us, Brazil, and I'm pretty sure that Colombia as well, which is what's been exposed during the pandemics, right? The COVID, what's going on basically? Why we don't have a vaccine, our own vaccine? That's the question is been bugging me a lot, and that's why I decided to write down these slides of the COVID-19 pandemics here for all of us. We can talk about it. I want to pick your brains on this as well. So basically, what happened was chronic lack of funding from basic research. This was going on in Brazil, and this limited our science-based action. So how can we make kits, ELISA kits, or molecular assays quickly if our laboratories don't have any funding for many decades? So basically, we are dry, and if that's so, it means we had to buy billions of dollars of molecular assays and kits from Korea, China, and everywhere. Um, all the decisions that we made here um, were based on data from other places, different cultures, different genetic background, and still we're making decisions based on this data, and we don't have data, local data. So this is a challenge that we have to face it. What the other things that we realize that we don't have the good manufacture uh, uh, GMP facilities or good manufacture practices. We need those. If you want to get on top of the production, you have to have those facilities. Basically, you don't have apparently here in Brazil. We have a few. The other thing that's important is that we have to have the structures to design and to make clinical trials quickly. And this goes with the regulatory agencies, in our case, Anvisa, which is not prepared for clinical trials from Brazil. Can you imagine this? It's amazing how fast a company or how fast a different international foundation country come here, ask for a clinical trial, they are approved quickly. And if when we ask, I don't know what happened. So as it works like people don't want us to make our own clinical trials. Can you believe that? Because all the things are easier for outsiders. So basically, there's this thing that the FDA said, if the FDA approved, all the regulatory agencies can approve, no problem. No way, right? So that's another problem. Uh, the other thing that we realize, we only have few production sites. In our case, Oswaldo Cruz Foundations and Butantan. And they are producing vaccines prepared or generated in other countries. So what I'm saying is, if we have all the people, we have everything here to make a vaccine like the inactivated SARS-CoV-2 vaccines, but we don't have it. We are producing vaccines prepared from different places. So of course, everybody wants a vaccine as quickly as possible. It doesn't matter who made this vaccine, right? Of course, I want a vaccine. I'm holding one vaccine from overseas. Of course. So basically, if I'm putting this slide here, a lot of people say, "Oh, you're crazy. What are you complaining?" No, I'm complaining not for today. I mean, for today as well, but I'm complaining for the next pandemics, which we're going to have. We're gonna have a next pandemic. Hear me out. What are we going to do? The opportunities that I see, I'm finishing, is that cooperativity. So that's the time that we can cooperate with each other. Also, we can implement local basic research and innovation, right? So if we're making now molecular kits for diagnosis, why not we can make for other things that's going on locally at our countries? And also, I think we are emphasizing that we have to have a scientific basic government. Science and government, they are as one. There's no division. People need to understand that. The generations need to understand that. Otherwise, we're gonna go down, as we are seeing here with the president of the Brazil, of Brazil, which from the beginning he starts being against vaccination. He starts to generating fake news for the Brazilian people. And we have many deaths, which is on his hands right now. And we need surveillance strategies. Since you're gonna have another pandemics in the future, we have to be prepared. We have to be able to develop algorithms to predict which virus will turn up, or which bacteria or resistant bacteria will arrive among us. And I also need to thank all the funding and support that we received from the Brazilian government, from the international institutes, and from our graduate program, the pharmacology. And again, thank you very much. Oh, that's the Praia Mole, the place we love. And Jair and Ibeth, I know a lot of the Praia Mole. And thank you very much.
Thank you, Professor Báfica.
Thank you.
So
Thank you very much, Dr. Báfica. Excellent conference.
Let me stop sharing.
Molto obrigada por la palestra. Excelente palestra. Jair, ¿tenemos preguntas?
Yes, Professor Nubia, thank you. Professor Báfica, thank you so much for your presentation. I think that was awesome. The students will be happy to know different concepts, and in the way that you explain us was awesome and excellent. Thank you so much. We have a question in the chat, and I don't know if the Professor Daniel want to open your microphone, but here we have the question of the Professor Daniel. He said, excellent conference, Dr. Báfica, thank you very much. Being the virus synthesized in a human cell, some people suggest that beside the spike protein in COVID-19, the glycosylation pattern of the virus after being delivered from the cell is also important. And such pattern may have a negative impact on the success of the vaccine. What do you think about it?
Thank you for the question, Danielle. I think that's an amazing question. Actually, we can discuss from the immunological perspective. So I'm going to start with this. All the vaccines developed so far, all different strategies. We have adenovirus, mRNA, inactivated. We have also the purified protein in trials, right? All of them are giving protection, at least efficacy, against the original virus, right? So in terms of the human immune system, when we're giving them some agent, could be only the spike information or the whole virus, like the coronavac, the Chinese vaccine, we're giving protection. So there's efficacy, at least what it's been reported in these papers. If that's true, it means that we can stimulate our immune system to make immunity against the virus, right? To at least some immunity. I mean, immunity that's protecting. But whether the glycosylation pattern it's causing the negative impact, we don't know. To do that, we have to have at least a model, a vaccine or candidate without the glycosylation and see what's going on, right? maybe you have some targets that we can try out without these glycosylation patterns, but we have to keep in mind that when we do that, we are going to stimulate very specific antibodies and TCRs that is raised against a place that is not glycosylated, sorry, which means that these the immune system would raise the response against a different part of the virus that you're thinking. So I think that we need to have the glycosylation in order to be fully protected. So I mean, we need to give the immune system what the virus is. But I think what we need to do is to find the right place or the right cell, let's say the mucosal immune system to stimulate. That would be my hunch on this, right? But I think these experiments that you're proposing have not been done yet, but I think it could be.
Okay, thank you, Professor. Here we have another question of Professor Daniel that he said, which may be the effect of being vaccinated with a vaccine produced with a strain from outside of our countries instead of the strains that are circulating in our countries.
We have to do that. I mean, we wish we could do that experiment. We cannot. But so we can think on two scenarios, right? The first would be that the HLA, right, the genetic background that's important for the antigen presentation, the human leukocyte antigen, I showed you before as MHC, the major histocompatibility complex. So the HLA circulating in our genetic background is different. So it's different in each country, right? Mm. When you get a virus that is circulating out there, it means people are making the response against antigens that might be relevant, epitopes that might be relevant for that response. When you get a virus from overseas, you could lose that response because the epitopes are going to be different. So the scenario one is you lose that response. If you lose that response, though, so we go to HLA and we have to look at that first. If you don't lose that response, it means that some epitopes are very conserved among human populations, and it can fit in any HLA. That would be a set scenario too. But yes, we have to keep an eye on this, and that's why we also to make we have to have data, local data, with our own people to have the vaccines and tests and everything and so on. But that's a good question.
Okay, thank you. And Alba Rodriguez said, Dr. Báfica, which vaccine do you consider to be the best for Latin America?
Amazing. I think we have to make our own vaccine. Whatever it is, we have to make it. Which one? Whatever we make, we make. Whatever is there's, and outside's outside. But we have to be on top of it. We need the data. We need data. We need to build together our own vaccines. I know it sounds crazy saying like that. I know. I can see that sounds crazy. But it's not the crazy way that I'm talking about. I'm talking about more of a free crazy way. Right?
Thank you so much. Professor Sonia Ospina said, thank you for your excellent talk, Dr. Báfica. what do you think about thrombosis present by some people, especially I think for AstraZeneca vaccine?
Yeah, we don't know what's causing. Nobody knows yet. There's been some reports. I think we basically don't know. I cannot think because I don't know. But the thing is, I invited a specialist, an expert in thrombosis, which is studying COVID thrombosis or with the tissue factor. He understands a lot of the tissue factor that it's expressed. Apparently, tissue factor is a protein that expressed in tissues in many cells, and this is the protein that induces the thrombocytic events in people. So this guy is an expert on tissue factor, and Ivo Francischetti, and he's going to give a seminar here in our department. He's in Maryland, USA right now, and I'm gonna send you guys the invitation. I'll send Hai. Hai can send you guys the invitation.
Thank you
right?
so much.
So, and you guys are all invited to know more about thrombosis. But Sonia, thank you for your question. I cannot answer that.
Okay, thank you, Professor, for the invitation. We are waiting for your invitation to connect to that talk. And Luisa Fernanda Velasquez said, thank you for your excellent presentation. Do you think current vaccines need to be improved?
For great question. So the first vaccine, COVID, let's say it's the first COVID-19 vaccine generation, right? I'm gonna say first generation now. So the first generation, they were designed to be first quick and also to block COVID-19 symptoms. Basically, that's what the whole thing people wanted to do, just decrease the number of people going to the hospitals. They were not worried about whether the vaccine would have an impact on viral transmission, so decreasing the viral loads in people. So we have two pieces of data showing that the AstraZeneca vaccine and the Pfizer vaccine in a few patients decreased the viral loads in patients that had COVID, symptomatic COVID. So people got vaccinated. They still had COVID, but moderate or light COVID, and they were looking at the swab to see the viral loads in these people, and they showed a reduction, which means that it's possible through vaccines to reduce transmission. That's why I think we still need improvements, because we have to have a vaccine that clear. So that's the main goal. A vaccine that blocks completely the viral transmission. So if you're vaccinated, you will not be able to host SARS-CoV-2. That will be the main goal. So that's why I think we need improvements. If you're going to reach that, I don't know. I hope so. Let's see.
Okay, Professor, we have another question. Say Lisette Acosta, how convenient is to vaccinate people who is immunosuppressed
Yo puedo leer esa acá. ¿Qué tan peligro es vacunar al paciente inmunosuprimido con cualquiera de las vacunas para COVID? Todas as vacinas, porque não tem vacina porque é o seguinte, o problema do imunossuprimido, pode falar em português? Eu não sei.
Pode.
Nos imunossuprimidos, o maior problema é quando você tem uma vacina viva, atenuada. Porque mesmo sendo atenuada, no imunossuprimido, esse micro-organismo ele pode proliferar. Então, isso é um problema. Como não existe nenhuma vacina para COVID até agora aprovada que seja atenuada, viva atenuada, então todas, a priori, elas são bem seguras para os imunossuprimidos, né?
Okay.
Agora, em ordem de segurança, a primeira delas seria uma subunidade, subunit vaccine, like a Novavax está fazendo. Então, seria só proteína. A segunda delas seria a mRNA vaccine. A terceira delas seria o vírus inativado, como a Coronavac. Então, elas estão tranquilos aí, eficazes para imunossuprimidos, seguras.
Okay, thank you. Yvonne said, thank you for your presentation. What do you think the vaccines are really the solution for the pandemic?
Thank you for your question, Yvonne. I think yes. One of the solution for now, I mean, scenario one, we have a vaccine that clears, that blocks viral transmission. Esse é o cenário one, the goal. Sim, vaccine per se will solve the pandemics. But we are not working this scenario yet. We are working in the real scenario right now. The one that we call reality is the vaccines, they are diminishing the COVID-19 disease, the symptoms, severe, moderate. The virus is still circulating. Even with the people being vaccinated, the data suggests that the virus being circulating. Although low, it could be lower virus, but it's still circulating, right? So we still have to add up all the things that we're doing, wearing the masks, the right masks, not going to the supermarket, the places that have been occupied by lots of people, like we cannot go to concerts, close restaurants. Yeah? So even with the vaccine, even if you are vaccinated, you still have to maintain the same practices. So because we don't know what's going on with the viral transmission. I mean, of course, you can now walk outside if you're vaccinated, but wearing the mask and glasses, not touching anybody, alcohol and all that kind of things that people are saying.
Unfortunately, Professor, we don't have enough time, but I want to ask another question. And what are your experience interacting with the pharmacological industry now that you are thinking to produce a vaccine?
No experience yet, but we are, so what we'll be discussing here is that we are going to apply for startup. If you have, so what would be my suggestion? Everybody in a lab, like let's say in a biotechnologia institute, would build together an approach, a new approach for a vaccine. And maybe you guys could think on revaccination. That's the thing, right? Revaccination to protect against the mutants or the variants that is going to circulate each year. Thinking on this, and you do a new thing, and then you make a startup.
Okay.
With the startup in hands, you guys have I don't know how it works in Colombia, but that's the way it would be here. with a startup open in your technology, and then you can hook up with the industry to make a clinical trial.
Okay.
Okay.
But for the clinical trial, somebody has to work at the government level to make that happen, right? The clinical trial is important. So you also have from the start, hook up with people that do clinical trials, and you do clinical trials. So you basically, in my experience, we have to cooperate with plenty of people, and we have to divide it up the work between us, thinking on us. Us, what I mean is the community, not thinking on ourselves, on our curriculum or anything, or papers or anything. We have to be thinking about the community. If that's the real deal, that's what we really want with the vaccine, we will be able to be free and not be in the hands of political gains or capitalistic gains from outsiders.
Yes.
Okay.
Dr. André Báfica, on behalf of all members of our community, we want to thank you for serving as a speaker in our institutional seminar today. I think it was amazing and put together to allow to think about the current problems in development of vaccines. And also thank you to allow us to know your advancing in your research about COVID vaccine development. And I hope that we can realize future collaborative works in a near future. Thank you so much, Professor. And Professor Anubia, do you have to say something?
Thank you. Thank you very much, Dr. Báfica. It's an excellent palestra. We hope other opportunities for we have in this space, in this seminar, and other possibilities for collaboration with us. Okay, thank you very much for this.
Thank you so much. Muchas gracias a mis amigos de Colombia, de Bogotá. Muchas gracias. E eu gostaria de deixar aberto aqui nosso laboratório para colaborações, para o treinamento, para o intercâmbio de estudantes, a atmosfera científica proporcionada por indivíduos de outros países, outras culturas. Essa atmosfera é a que nós gostamos aqui no nosso laboratório. Nós temos muitas pessoas de vários lugares do Brasil, nós treinamos também pessoas de vários lugares do mundo, e todos estão convidados. Vocês podem visitar nosso site, mas todos vocês estão convidados a mandar um e-mail se tiver dúvida ou qualquer coisa, nós estamos aqui. Muitas graças, muitas graças. Bom ver vocês, bom também saber que o Instituto contratou o Raí, a IBET também está associada, é bom ver vocês crescendo e contribuindo para a comunidade. É um prazer.
Muito obrigado, professor.
Muchas gracias. Muchas gracias.
Tchau, tchau.
Para todos, muchas gracias a todos nuestros asistentes. Están cordialmente invitados para nuestra próxima sesión, a nuestro seminario institucional. El día 15 de abril tendremos la participación de la profesora, de la doctora Luz Estela González de Bachán, que es investigadora en el Centro de Investigaciones Biológicas del Noroeste Mexicano. Entonces, todos cordialmente invitados. Muchas gracias por su participación. Muy amables. Que tengan todos muy buen día. Y a cuidarse, por favor, todos.
Gracias, profe Nubia.